So today I'm going to talk about our experimental method to map microRNA targets transcriptome-wide in single cells. So it's not so computational, but there's a lot of computation involved also. So I hope you'll find it interesting. And I mean, everybody here knows about microRNAs, but I'm just going to very briefly outline a couple of features that are relevant to my talk in particular. So microRNAs are 22 nucleotide long RNA molecules that can repress, post-transcriptionally repress the expression of protein coding genes. They function by guiding the Argonaut effector proteins to their target, so functioning as a kind of sequence-specific adapter. They're present in all multicellular animals in numbers that roughly scale to the complexity or number of cell types of the organism. So nematodes have around 200. And while people in the audience here could also probably very hotly debate the number of microRNAs in humans, let's say that they're at least close to 1,000, and we don't disagree too much. And they have important roles in many different biological processes, including formation of cell identity development and in human disease. And through studies of sequence conservation, it's been estimated that upwards 60% of all human genes are regulated by one or more microRNAs in one or more cellular contexts. So it's a very prevalent kind of regulation. And importantly, each microRNA can target hundreds of distinct messenger RNAs. So every microRNA obviously has its own repertoire of targets. And to understand what a microRNA does, we need to unravel what are these targets. Because, as I said, the microRNAs function as a kind of adapter between the messenger RNA targets and Argonaut, which actually does the heavy work. So how do we identify microRNA targets? So since 2003, researchers have been identifying targets computationally. And the method of doing so has changed pretty little over the last almost 20 years. So we know that for the microRNA, the nucleotide 2 to 8 are particularly important, and this is called the seed. So what people do is that they align genomes, then look at the primary DNA genome sequence and look for these kind of conserved seed sites in 3' UTRs. And on the upside, if you find a binding site that is very deeply conserved, it's also likely to be actually functionally important. So not just that there is an interaction, but it's actually there's actually selection pressure to maintain it. On the other hand, since this is essentially a computational analysis on primary DNA sequence, we know that it has some limitations. All of these methods have fairly high false and negative positive rates, and overall, the accuracy of these methods have not changed, let's say, very radically since 20 years ago, although they're still very useful. And also, they're not specific to a particular tissue or sample of interest because they just analyze the DNA sequence. So the state of the art on the experimental side is agoNOT-CLIP. And in this method, you take cells, you take your tissue of interest, cross-link proteins and RNAs using UV light, then lyse the cells. You restrict then the RNA fragments so that you have only the ones that are essentially covered by proteins. And then using immunoprecipitation, you pull down Argonaut specifically, like it on adapters to the messenger RNA targets, and sequence them using NGS. And in this way, you get kind of nice peaks that can be used to identify which regions were interacting with Argonaut. And so on the upside, this is a genuine experimental detection of target sites in your tissue of interest. And it's been shown that sometimes this causes nucleotide conversions, meaning that you can identify the binding sites with nucleotide resolution in some cases. However, you can imagine that Argonaut actually spends a lot of time in the cell scanning for functional targets. And these spurious scanning interactions are also detected. and give pretty clear signals. So even though you maybe detect a genuine interaction between Argonaut and an RNA in your sample, this doesn't mean that repression takes place. And also the method is very experimentally demanding. So you need good antibodies, you need to UV crosslink, you need gels. It's a very long protocol that can take upwards, I think, 5-6 days. And also importantly, you require typically in the order of millions of cells to actually do this. And in my lab, so we're funded by an ERC grant to look at microRNA functions in single cells. So obviously this won't do, that you need millions of cells. So we've been looking for a way to do something like ClipSeq, not necessarily better, but something like ClipSeq, but in single cells. And how could we do that? So a couple of years ago, we saw that there's this new method called agoTRIBE, developed by the Rosbash lab. And the principle is quite simple and it's very promising. So you have here in A, an RNA binding protein of interest. This could be Argonaut or HUR or whatever. And then you're leveraging on the ability of this naturally occurring ADAR protein to edit RNA. So the ADAR proteins, they have double-stranded RNA binding domains, and then they have catalytic domain, which causes deamination. So adenines become converted to inosines, which are then detected either in sequencing or the cell's natural machinery as guanine. And this in the cell, for instance, can be used to change amino acids. It has many different functions. It's a little bit like a Swiss Army tool of cells. And so in agoTRIBE, you fuse then the RNA binding protein of interest with this catalytic domain. And the idea is then that the RNA binding protein guides this fusion protein to the natural endogenous targets. And then the ADAR domain causes this conversion that can then be detected in sequencing. So you see, you don't need actually really any immunoprecipitation or any highly lossy steps. So what we've been doing is exactly this. So we fused the deaminase domain of ADAR2 to Argonaut. We transfected this construct into hex cells, and then we've applied bulk or single cell sequencing to look at where we find these A to G conversions in the resulting sequencing data. So the first thing we wanted to see is where does our fusion protein localize? Does it localize similarly to naturally occurring Argonaut? And so you have here GFP tagged AGO2 and here GFP tagged agoTRIBE. And you can see that the localization is very similar, and in particular the overall pattern. And interestingly, you see these little bright green spots, which could very well be P bodies, which are the processing bodies where microRNAs relocate their targets. And so also without GFP tagging, but using antibodies against natural ADAR2 or AGO2. So we did basically co-imaging, and we see here again quite a lot of co-localization in this orange. And we see that once we transfect agoTRIBE, we have a lot of spots where AGO2 and ADAR2 are co-localizing in something that could be P bodies, and we speculate that this is probably the fusion protein that localizes there. So, but we think that our fusion protein should be then doing these A to G conversions. So do we actually see this? So we did RNA sequencing on 50,000 hex cells. And you can see here the 12 different types of nucleotide conversions that exist. And most of them stay very constant. We have here controls. Here we have some pretty strong controls, which is the ADAR only. So this is basically only the ADAR deaminase domain, but without Argonaut and without microRNA. So this gives us some kind of background of just transfecting in ADAR by itself. How much editing does this cause? And we can see that from the controls to ADAR only, there's an increase, but even more with agoTRIBE. And in orange, you can see that these are specifically the A to G conversions that we expect. So where does this editing occur? So you can see that for a lot of different annotations like long non-coding RNAs, pseudogenes, introns, this changes very little. On the other hand, for the parts of annotations that are part of mature messenger RNAs that we expect would be the actual targets of microRNAs, we see that this increases very strongly. So 3' UTR and CDS both increases very strongly when we transfect in agoTRIBE. And here you can see it as a percentage, that the kind of more nuclear annotations that we expect shouldn't be targeted by Argonaut or agoTRIBE are depleting. So we worried, however, that maybe just by transfecting in a lot of ADAR2 into cells that we could cause a lot of unspecific editing. So you can imagine that ADAR2 might be just bumping into things at random in the cytosol. And so to control for this, we compared editing levels. So every dot is one gene. So we compared editing levels between our agoTRIBE transfected cells and the ADA-only transfected cells as a control. And we see that some genes are fairly specifically edited in the ADA-only, and we think that these are the kind of spurious editing events that we're not interested in. And then we saw that many genes or many transcripts are edited highly specifically in agoTRIBE and not when we just put in ADA by itself without Argonaut or microRNAs. So we took the top around 1,000 of these and putatively assigned these as our agoTRIBE targets. This is still in 50,000 cells, just to keep you all on track. And then we compared this with the targets of clip methods, of 3 state-of-the-art clip methods, HITS-clip, or that have all been used, HITS-clip, PAR-clip, and E-clip. And we did this Venn diagram. And it's a little bit difficult to maybe see exactly what's happening there in the middle. But I think one thing you'll appreciate is that every method has a number of targets out of the top 1,000 that are unique to it. And so you would think that if there's one method that's very different from the rest, it would have many more unique targets. And in this case, if we do a kind of Pepsi test, so imagine that you can't see the labels, which is which, and you'd have to guess which one of these is the non-clip method, you would actually think it would be this one because it has the most unique targets. But in fact, this is E-clip. So in a sense, agoTRIBE is more like a clip method, like the other clip methods, than E-clip is in this respect. And here again, we did the same thing, but just with a Jacquard similarity index. And you can see the methods that resemble each other the most in the target repertoire is PAR-clip and HITS-clip. But agoTRIBE is actually fairly close in similarity. And again, E-clip is a little bit like an outlier. So it looks from this point of view that agoTRIBE is really behaving like a clip method in its target selection, even though it's not actually a clip method. So again, we worried that maybe the reason we see this overlap between the clip methods and agoTRIBE is that both methods has the same kind of biases towards, say, highly expressed messenger RNAs. So to correct for this, we compared the overlap of HITS-clip targets with, on one hand, our agoTRIBE specific targets, and on the other hand, the ADA-only specific targets. So these are the ones from our controls that you remember. And here we see a fourfold enrichment in the overlap in the agoTRIBE compared to the ADA-only controls. So what this really suggests is that the reason we have this overlap is because of the ago part of TRIBE, so that it's really microRNA guidance that causes this nice overlap and concordance between the clip methods and the agoTRIBE. And so I mentioned a little bit earlier the computational methods to look at the microRNA targets like target scan. So of course, we also wanted to overlap with that. And we see here an overlap between the top target scan targets and agoTRIBE targets of 112. and this is 3 times higher than if we look at the ADA-only control. So the reason why we have this overlap with the computational microRNA target predictions is because of exactly the AGO part, the microRNA guidance, and not just that we have a lot of random editing. And in fact, we have a better overlap with target scan than does hits clip, which is again pretty comforting. So last time I presented this talk, I got the feedback, okay, but how good is agoTRIBE? And digging a little bit into it, I found out that what the audience wanted to know is, so the agoTRIBE targets, are they functional? So we did a functional experiment also, where we, in a separate experiment, so this is actually separate from the agoTRIBE transfection, we inhibited microRNA repression with a small protein called T6b. And what T6b does is that it binds to the pocket of Argonaut2 where TNRC6b normally binds. And this is a protein that's necessary for microRNA repression. So when this gets displaced by T6b, the hypothesis is, or the evidence is that Argonaut is still binding, but it cannot repress anymore because it's not interacting with the TNRC6b. And so we see here, looking at the target scan targets, we see a shift, which shows a de-repression. So from this evidence of the predicted targets, we think that the experiment basically worked. So what about now then our agoTRIBE targets versus hit clip target? How are they de-repressed? And we see that both our agoTRIBE targets and also the hit clip are de-repressed like 0.18 in a log-fold change, so not very strongly, but something that's consistent with literature. And when we only consider the ones that have a conserved seed sites, this increases. So what we can say is that the agoTRIBE targets are actually functional, or at least as functional as our clip targets. And this is what we wanted. Okay, so I promised at some point that I was going to talk also about single cells, and this is now the next part of the talk. So to profile editing in single cells, we decided to use SmartSeq3. And this is a method that doesn't have the same throughput as does, say, 10x, for instance. So it doesn't do millions of cells, but it's very, very sensitive. And also it gives very good coverage of the whole transcript bodies. And this is something that's interesting because we want to capture at least the whole 3' UTR. And so we transfected this into 703 hex cells, and then we had 26 control cells. And so last time I presented this, another comment from the audience was, okay, but how do you control for the difference in transfection levels? When looking at individual cells, it's quite important to know, is editing not occurring in this cell because microRNAs are not targeting anything, or because the cell was initially just not transfected by agoTRIBE? And so we figured out a way to estimate transfection levels. So you remember that we have ago2 and the ADA2 deaminase domain, and then we have an artificial linker sequence between here that, to our knowledge, doesn't exist in nature. So since we did RNA sequencing of these single cells, we also detected actually the construct, the RNA construct, and we could then map against this, map uniquely against this synthetic linker and use the linker counts as a kind of proxy for transfection levels. And you can see here that in agoTRIBE cells, we have a lot of these linker reads. This is summing over the 703 agoTRIBE cells. And in the control cells, we have very few. So this is added a pseudo count of 1. So most have 0. And then there's some that have a few, and we think these are mapping artifacts or index hopping or something like that. And regarding editing levels, we also see big differences. So In the control cells, we have typically around 1,000 editing events, because keep in mind that these cells actually do have endogenous ADA also, just at very low level. And then for the transfected cells, we have around 10 times more on the average, so 10,000. And when we plot these against each other, we have here the normalized number of linker reads as a proxy of transfection efficiency. And here we have the number of editing events. So we see that the control cells, they are the dark gray triangles, and they typically have essentially no linker counts as expected, and very low editing levels. And some of the agoTRIBE transfected cells behave in the same way, suggesting that they were not transfected efficiently. And then we have a lot that are behaving nicely. And so basically what we can do here is to do a kind of computational gating, a bit like cell sorting, where we say, we put some cutoffs here and we only consider the cells that have, for instance, more transfection or more editing than at a certain level. And in this way, we can avoid the problem of dealing with some cells that were not efficiently transfected. And we also wanted to look at how much does this transfection actually, because it is a perturbation, right? How much does it affect the transcriptome of the single cells? So you can see here the control cells, and here the agoTRIBE transfected cells, which are overlapping, but in part distinct. And then we compared that with basically another lineage of hex cells. So it's basically still hex cells, but these are fast growing, so at like a different batch. And you can see that they group separately from the rest here. So suggesting that, yes, we do see some differences because of agoTRIBE, but they're comparable to the difference between two similar strains of hex cells, so quite subtle. So looking then at single cells. So we had here one target, SBN01, which is known to contain 3 miR92 binding sites, and this is a microRNA that's highly expressed in hex cells. And you can see here a representative control cell and a representative agoTRIBE cell. So you can see that even though these are just single cells, we get a lot of data from the sensitive SmartSeq3 just for this particular 3' UTR. And you can see that in the control cell, there are virtually no editing events in yellow, but while this particular UTR is just covered in editing events in the agoTRIBE transfected cells. So you can see this is a kind of really fine resolution that we get with these sequence data. And we next thought, okay, let's apply this to a biological question. So not very much is known about microRNA targeting across the cell cycle. So we took our cells and used SOIRAT to assign them computationally to cell cycle stages. You can see this here is actually, I think, UMAP, where we then placed a kind of graphics around saying this is S phase, G2M, and G1. And we sorted the cells computationally into numbers that correspond quite a lot to the relative fractions that have also been described in the literature for hex cells. And we next wanted to see how much does RNA levels and microRNA targeting, like as in total microRNA targeting, change over the cell cycle. And we could make this estimate because we normalized our pseudobulk measurements to spike-in sequences. So we put in artificial sequences in every cell and could use this to basically normalize over and estimate absolute abundances. And we can see that overall RNA levels appear to go up from G1 to S and stay relatively constant to G2M. And this maybe makes sense because we know that over G1, there's transcription taking place, and then starting S, very little. And in contrast, the kind of total pool of microRNA targeting as estimated by editing seems to increase linearly through the entire cell cycle. And we have two different, we find this really intriguing, and we have two different hypotheses as why this might be. And the first one being that it is known from the literature that at least in some mammalian cell lines that there are just more microRNA molecules in cells in the later stages. So that could reflect that. On the other hand, it's also possible that it's a kind of cumulative of microRNA editing that is occurring over the cell cycle. So we have agoTRIBE just transfecting, just editing more and more transcripts. So it's a kind of molecular clock of the accumulated microRNA targeting that has occurred. And then at G2M, we have cell division, and then following the cell division, we have G1 again with a burst of new transcription, and then we have basically all of the accumulated targeting as evidenced by editing being kind of diluted out. And we think that both hypotheses are interesting and important. And now we're looking into it by also looking at half-lives. So basically seeing, is this tendency higher for transcripts that have higher half-life, which would then favor the molecular clock hypothesis. On the other hand, if we see that this tendency holds for all transcripts, irrespective of their half-life, then we think it's more about that there actually are more microRNA targeting happening in the later cell cycle stages. So we can also do this at the level of individual genes. So we have these measurements and these curves of, on one hand here, gene expression and gene targeting by microRNAs for, well, around all of the 10,000 genes that we can reliably measure. And we find that, for instance, this CCNB1 is important for the transition from G2 to M. So we expect it to increase in expression at this particular stage. So it's not targeted by microRNAs very much. This one is known to, is also a cell cycle gene known to facilitate transition from G1 to S phase. And we see it peaking exactly where we expect it to. And we also see some genes with very interesting patterns. Like, for instance, this SENPA is a centromeric protein, which appears to be very important during cell division. And it seems that this is very heavily targeted in G1 and S phase, and then microRNA repression is alleviated in G2 to M. And this basically fits well with expression patterns that's very low in G1 and S, and then goes up very highly in G2M, so suggesting that there's some kind of de-repression in part. So we have many of these very interesting patterns, and this is just one example of what the method can be used for. So to go a little bit into the outlook, so of course, people talking about single cell methods, they will always tell you about heterogeneity, and I'll do exactly the same to not disappoint. So of course, when we do bulk RNA sequencing, or let's say bulk Argonaut-CLIP, then we get a big pool of targets over a cumulative of 1 million cells, right? But what we don't know is within this pool of cells, are the same microRNAs targeting the same targets, or do we have different microRNAs targeting different targets? So it could be very heterogeneous and very complex patterns of targeting that's basically obfuscated when we do CLIP-seq to profile 1 million cells. So of course, what would be very interesting would be to look into a heterogeneous cell culture. Could be, for instance, embryonic stem cells being differentiated into neurons, and see what's really happening at in the individual cells during this dynamic change. And also, are there some clear tipping points that we can identify when doing all of these cell projections and velocity projections? I think it would also be very interesting to look, for instance, at organoids in complex tissues, like representing complex tissue, saying, okay, do we see, what kind of targeting do we see in muscle cells? What kind of targeting do we see in brain cells, in organoids? And of course, the dream is also to do this in animals at some point, right? Imagine making a construct where we can express agoTRIBE in mouse brain, and then we can look at differential miRNA targeting in different, not just regions, but in different individual cells of the mouse brain. And keep in mind that we don't need for this purpose to actually physically sort out the different neurons, because we can computationally assign the cells to the different tissues and do pseudobulk analysis, or even single cell analysis. Okay, so since I'm the PI, I didn't actually really do this work, obviously. So I'd like to thank the people who did do the work, in particular Ina, who was really driving the experimental work, designing the fusion protein, testing, redesigning, getting it to work. Also Vaishnavi V, that I think is listening in, who did the vast majority of the computational work, but also with very substantial input from Panas and Emilio. I'd like to thank our collaborators from the different groups, and also the facilities here at SciLife Lab, and our funding buddies. And finally, thank you for your attention.